Okay, I think we should get started. I'm David Spector. I'm Director of Research at Cold Spring Harbor Laboratory, and it's my great pleasure to welcome you to the laboratory and to tonight's lectures on immunotherapy and cancer, the latest research. Cold Spring Harbor Laboratory is one of 69 National Cancer Institute designated cancer centers throughout the United States, and the laboratory has been a leader in cancer research for more than 40 years and has a very long history of both innovative discoveries and major breakthroughs in cancer research. A little over 2 years ago, the laboratory entered into a strategic affiliation with Northwell Health, bringing together their state-of-the-art healthcare system and large patient population with our world-class scientists. And together, we hope to move basic science being conducted here at Cold Spring Harbor Laboratory to the clinic more rapidly to help patients directly. And we have a lot of exciting collaborations going on together with Northwell Health. And I'm sure that over the next few years, you're going to hear a number of lectures from scientists, both from Cold Spring Harbor and Northwell, who are working together to make new advances and better treatment options for individuals with a variety of cancers. This evening, you'll be hearing from two outstanding clinician scientists on a topic that has been in the news quite frequently, in fact, on the front page of the New York Times yesterday. This approach takes advantage of your body's immune system to launch an attack on cancer cells. And so it's a very powerful approach. And tonight you'll be hearing about how this approach works, what the caveats of this approach are, and how these issues can be improved to make this approach more valuable to a larger population of patients. So our first speaker this evening is Professor Doug Fearon. Doug received his MD degree from Johns Hopkins University School of Medicine. He's been a professor at Harvard Medical School, Johns Hopkins University, and he's been a group leader at Cancer Research UK in Cambridge, England. He currently holds a joint faculty appointment as professor at Cold Spring Harbor Laboratory and Weill Cornell Medical College in New York City. His laboratory has made major contributions to various aspects of immunity, including cancer immunotherapy. Doug has received numerous awards, including being a member of the National Academy of Sciences, a fellow of the Royal Society, and a member of the Royal Society of Physicians. And so it's my great pleasure to introduce Doug Fearon to give the first lecture this evening.
Thank you, David. David's kind of given you my background, and I thought since this is a somewhat informal occasion, I would sort of relate the curious nature of my research career. As a clinician, I was a rheumatologist, and I was dealing with autoimmune diseases. And of course, in autoimmune disease, you're trying to turn the immune system off because the immune system is damaging normal tissues. And in an attempt to do that, I felt I needed to know more basic immunology, so I became more and more of a basic immunologist, and it's actually been some time since I've seen a patient. But as I learned more and more about immunology, it actually became apparent to me that perhaps it's easier to turn the immune system on than it is to turn it off. Once the immune system decides it wants to attack something, it sort of hardwires that. It's what we call immunological memory. So it's hard to turn the immune system off. And that realization occurred at a time, and I was serving on a scientific advisory board of a cancer research foundation. And I said, oh, in cancer, we want to turn the immune system on. And so that led to a change in my career, about 10 years ago, and I became interested in cancer immunology. And so I'd like to discuss with you today Cancer Immunotherapy, a rapidly evolving clinical science. I apologize for a slide having lots of words on it, but I'd like to at least logically break down the talk into 4 elements. The immune system can control cancer because cells in the immune system called T lymphocytes, or T cells, have the ability to kill cancer cells. They have this ability because cancer cells invariably, almost invariably, we think, perhaps look like they're foreign. And as you all know, the immune system can reject foreign tissue, an ability it has because this is the way it actually attacks virally infected cells. So really, we're not asking the immune system to do anything that it cannot do when we ask it to kill cancer cells. T cells have the ability to do that. I'm going to show you a really nice video done by my colleague at the University of Cambridge, Gillian Griffiths. Now, in order for T cells to do this, they must contact the cancer cell. They must contact the cancer cell in order to kill it. And in that contact, the T cell has a receptor that recognizes a structure on the cancer cell or a virally infected cell that triggers the T cell and elicits its killing activity. So I'm going to show you a video done by Gillian Griffiths, as I said, who is a colleague and a close friend at the University of Cambridge. I think this will show strikingly what the immune system can do, the lymphocytes. So this short video is of T lymphocytes in tissue culture. It has some dramatic music associated with it.
These are immune cells called cytotoxic T cells. Cytotoxic T cells are able to recognize and kill virally infected and cancerous cells in the body. These T cells are on patrol, searching for their targets. Let's take a closer look at how they move. Cytotoxic T cells are constantly migrating around your body. They move through your tissues by pushing out the membrane at the leading edge of the cell and pulling themselves forward. When a T cell encounters a cancer cell, an explosion of membrane protrusions explore the surface of the target.
This is when the T cell receptors are getting engaged on a T cell by
Cytotoxic T cells kill their targets with toxic chemicals. The chemicals are housed in these red structures called lytic granules. You can see the lytic granules polarized from the rear of the T cell to the blue cancer cell.
Those granules.
These toxic granules secrete specifically toward the target, protecting innocent bystanders which may be nearby. As the membrane of the target is compromised by the toxins in the granules, our indicator flashes bright red, signaling the impending death of the cancerous cell. Movies like these allow us to study the efficiency with which cytotoxic T cells operate.
And this is literally what will happen when we have a successful
Now
immune
we'll leave you
response
with a
against
few
the
more
cancer.
movies of T cells doing what they do best, killing cancer.
Okay. So that's an in vitro study, but we think essentially the same things go on in vivo when a T cell encounters a cancer cell for which that T cell is specific. Now, there are 2 ways that tumors escape T cell killing. And I think these are, have naturally evolved to protect normal tissues from T cell attack. The cancer didn't invent anything new. These were protective mechanisms that evolved to protect normal tissues from T cell attack. And the 2 ways are inhibiting T cell stimulation by cancer cells, and that it will inhibit the signaling that the T cell receptor imparts inside the cell to lead to secretion of the cytolytic granules. It's a signaling process, and there are ways that cancer cells have of inhibiting that signaling process. And the other way that tumors escape T cell killing is they can prevent the accumulation of T cells in the tumor, so-called T cell exclusion. So that's pretty straightforward. If a tissue wants to protect itself from T cell attack, it can do it by two ways, and maybe tumors do it by both ways, and some tumors do it by more one way or the other. You prevent the T cells from even getting into the tumor, and that obviously means you escape immune control. Or if T cells get in, you turn off the signaling through the T cell receptor. So current immunotherapy is directed at these two ways that tumors escape immune control by T cells. It's perfectly logical. And the current immunotherapy that you have been reading about and hearing about that's very exciting is ways to block the so-called T cell checkpoints, ways to block those inhibitory molecules that prevent T cell receptor signaling. Now, there are two scientists that have contributed to these approaches. Jim Allison on the left and Liping Chen on the right. I put the dates up here to let you know kind of what is the pace of translational research. Jim published his paper on a membrane protein on T cells called CTLA-4, showing that if you block the activity of CTLA-4 on T cells, you could promote tumor killing in a mouse model in 1996. It wasn't until 2010 that the first report of a clinical trial using antibodies to block CTLA-4 function reported that melanoma patients, about 10 to 15% of melanoma patients, had long-term cures, long-term remissions. So that's a 1996 to 2010 time span. In fact, the molecule of CTLA-4 was discovered by Pierre Goldstein in 1987. So the membrane protein in T cells, CTLA-4, was discovered 9 years before Jim discovered that it could have a role in T cell killing of cancer cells. Liping Chen worked on another protein on the lymphocyte surface that inhibits T cell receptor signaling called PD-1. And he published his first paper showing PD-1 was inhibiting T cell killing of cancer cells in 2002. Hanjo actually discovered PD-1 molecule in 1992, again a 10-year gap between the initial discovery of a protein and its understanding that it may have a role in cancer. And the clinical publications that antibodies to PD-1, so-called checkpoint inhibitors, was published in 2012. Now, I'm sure that some aspects of this we can speed up, but it may be an irreducible stupidity on our part to account for this slow progress. I just would let you know the logic of PD-1. So here's a T cell, and the T cell receptor is what recognizes a tumor cell, so-called MHC plus peptide. And that imparts a positive signal for activation of the cytolytic T cell, leading to killing of the tumor cell. PD-1 was engaged by a ligand called B7H1 on a tumor cell, actually leads to a negative signal that turns off T cell receptor signaling. And immunotherapy is an antibody called anti-PD-1, specific for the PD-1. It binds to PD-1 and blocks the binding to PD-1 by B7H1 on the tumor cell, so now the T cell receptor actually can be stimulated by the tumor cell and it can kill it. And to show you the glass that's half full, glass that's half empty, nature of clinical research. Here is the first reports of anti-PD-1 blocking, anti-PD-L1 blocking antibody or blocking antibody to PD-1 in 2012 New England Journal of Medicine paper, both from Johns Hopkins. Here are the cancer types, the number of patients, and the number of responders of those patients. So you can see melanoma patients respond to both anti-PD-L1 and anti-PD-1, about a fifth. Renal cell carcinoma, some patients respond. Non-small cell lung cancer, some patients respond. But so this may look like, not much, but these patients would have died of their cancers. There was no therapy for these patients. So this immunotherapy really was a dramatic advance for clinical oncologists. But then there are tumors that do not respond at all. Colorectal cancer, pancreatic cancer, prostate cancer. I have an asterisk for colorectal. There's a subset of colorectal cancer patients that respond. So our challenge is to try to figure out how can we get these tumors to respond. So maybe those tumors are not responding to so-called T-cell checkpoint inhibitors, which enhance T-cell receptor signaling, because these are tumors maybe the T-cells don't get in to the tumors. So enhancing T-cell activation isn't going to help because the T-cells aren't even encountering the cancer cells. So the challenge, at least from my lab, is developing immunotherapy to overcome this T-cell exclusion. So, and we wonder whether or not this is actually a more fundamental way that tumors escape immunity. If the T-cells can't be getting into the tumor, that's the most effective way to protect yourself from cancer killing. These are biopsies of human pancreatic cancer. The cancer cells are colored in red, and you can see the tumor is comprised of cancer cells and regions in between cancer cells called stroma. The T cells are stained, these are the green, whitish dots, these are T cells. And you notice that in this tumor, almost all the T cells are in the stroma, not in amongst the cancer cells. It's a non-random distribution. They're being excluded from the vicinity of the cancer cells. You see the same thing in this biopsy of a pancreatic cancer. This one doesn't even have any T cells even in stroma. But we know that occasionally, that can happen because occasionally you have a biopsy like this, where you have T cells in amongst the cancer cells. And you see the same phenomenon in colorectal cancers. Most colorectal cancers look like this, and occasionally about 10% of colorectal cancer patients will look like that. And that's because those cancer cells have many more mutations because of a genetic abnormality in the cancer cells. And the same thing happens in pancreatic cancer. And these patients do respond to checkpoint antagonist therapy, but none of these do. So can we overcome that T-cell exclusion? I'm not going to go into the basic research that led up to this approach, but we chose a target called CXCR4, which is a receptor that directs the migration of immune cells. And we had some evidence that CXCR4 signaling was somehow inhibiting T-cell accumulation in tumors. There's a small molecule called AMD 3100 which actually blocks CXCR4 signaling. So just to show you what preclinical research can do, this is a mouse model of human pancreatic cancer, actually developed by David Tuvison, who's here at Cold Spring Harbor. He developed it when he was a postdoctoral fellow with Tyler Jacks at MIT. And I've stained here, the cancer cells are staining in green, and the T cells are in red. And you can see in this mouse model of pancreatic cancer, T cells are excluded from the vicinity of cancer cells, just like you saw in the human biopsies. They're in the stroma, but not amongst the cancer cells. If we treat the mice for 24 hours with AMD 3100, this inhibitor of CXCR4, you see T cells amongst the cancer cells. And furthermore, this tumor now responds to the checkpoint antagonist anti-PD-L1, where it didn't here, and you actually combining AMD31 plus anti-PD-L1, you have elimination of cancer cells within 24 hours. Now we can objectively quantitate the redistribution of T cells with AMD 3100 by sectioning the entire cross-section of the tumor, staining it, and using a computer to measure the distance between each cancer cell in green and the nearest T cell in red. And you get a histogram. And this is the frequency of those cancer cells and their distance from a T cell. And you can see PBS-treated mice, the T cells are far away from the cancer cells. You do not alter that with anti-PD-L1, the red histogram, but with AMD 3100, you make the T cells come closer to the cancer cells. And now they're able to engage the cancer cells and potentially kill them, and that killing process can be enhanced with anti-PD-L1. So, what is the purpose of all this preclinical research? It is to say, is a mouse giving us a clue for what might happen in human cancer? So we're conducting a phase 1 trial of AMD 3100 in colorectal and pancreatic cancer to ask a question whether or not CXCR4 inhibition will promote the intratumoral T cell immunity. And this is funded by the Lustgarten Foundation, which many of you may know funds a lot of pancreatic cancer research and Stand Up to Cancer. I just would let you know that this cannot occur without a clinical research group, and these are the individuals at the University of Cambridge engaged in the clinical research, and this is my preclinical group here at Cold Spring Harbor. And I want to give you some of the results that we are getting in early results of this phase 1 clinical trial to allow you to get into my shoes and to say, you know, is there a hint? Is something going on? Is inhibiting CXCR4 in human cancers having the same effect that it did in a mouse? So the protocol is that we give 1-week infusion of AMD 3100 in patients with metastatic cancer disease, either colorectal or pancreatic cancer. We're limited to 1 week because it's just a phase 1 trial whose real purpose is for toxicity. We're in the dose escalation phase where we start out at a low dose and we have a target dose and we enroll patients at a lower dose. If things are all right, we do higher dose. If things are all right, higher dose till we get to the target dose. And so we're in the dose escalation phase, and all the patients I'll be discussing are 11 colorectal cancer patients. Now, how we're assessing what happens is we're taking biopsies of metastases in the liver before we start treatment and at the end of treatment. And we'll do various assays of those biopsies of tumor metastases to say, have we changed the immune reaction in the tumor? Our 1-week readout precludes a test of therapy. We do not expect to cure any cancer, which also means we're terribly grateful for these patients participating in the trial because it's very unlikely that they will receive benefit from their participation. We hope that benefit will come for patients in the future. And we ask the question, does AMD 3100 or is inhibition of CXCR4 enhance expression of immune-related genes in those biopsies? And so we measure the expression of immune genes using a technique called RNA-seq in the biopsies. And I'll show you some striking results. So the first thing that was sent to me by the team in Cambridge was a bioinformatic analysis of the genes that were upregulated with a post-treatment biopsy compared to the pre-treatment biopsy. And then you have programs to say, well, that pattern of upregulation genes, what does it most resemble? What biological process does it most resemble? And the top hit was allograft rejection, which is called a false discovery rate of 0.006, so 6 out of 10,000 times this will pop up by chance. Allograft rejection is precisely the biological process we want to get going in the tumor. T cells mediate allograft rejection, it's transplant rejection. So this came to about a month ago. That was terribly exciting. And for the other significantly pathways in which genes were upregulated was interferon gamma response and immune response, type 1 interferon immune response, IL-2 signaling, inflammatory response. So the treatment is promoting immune reactions in the cancer biopsies. One other thing. This is the level of RNA levels for the two chains of the T cell receptor. The T cell receptor on each T cell has an alpha chain and a beta chain. This is the way the T cells, so the number of T cells in the tumor will be reflected by the amount of message for those T cell receptors. And you can see some patients, I've circled, pre versus post-treatment, increase the message for T cell receptor alpha and T cell receptor beta. So we interpret that as potentially meaning there are actually more T cells in the tumor post-treatment with the AMD 3100, which we had predicted. And what's interesting about that is we can switch it and say, well, what genes are inversely correlated with expression of T cell receptors? So we have upregulation T cell receptors. What's downregulated in the tumor that have increased number of T cells? And what you have is a bunch of biological processes, all of which relate to cancer cell replication. Mitotic cell process, cell cycle process, et cetera. So this is inferential evidence that the increased T cells in the tumor are decreasing the number of cancer cells. It's inferential, it's a hint, it's a clue. And furthermore, these are all colorectal cancers, and it's interesting which tissues express these downregulated genes that are inversely correlated with increased T cell receptors is colorectal tissue. So it makes sense. Finally, I just want to show you one last thing. So we really pore over these data saying, what hints can we get? And this is another interesting correlation. It's a negative correlation between the fold changes of immune markers, and the two immune markers are called Fas ligand and granzyme A. And these are proteins expressed by the cytolytic T cells that actually kill cancer cells, Fas ligand and granzyme A. And what we've plotted here is keratin 19, which is a marker of the cancer cells. It's only expressed on cancer cells in the biopsies. And so you have decreasing keratin 19 with increasing Fas ligand, a negative correlation. The more Fas ligand message you have in a tumor, the less message you have for keratin 19, suggesting again that the tumors that have these T cell killer molecules are losing cancer cells. And the same thing with granzyme A. So I'll finish with that. Advances in immunotherapy depend on understanding basic immunology. You don't want to go into this guessing. You want to have some mechanistic concepts that you're actually testing. Jim Allison identified CTLA-4 as a target for immunotherapy in 1996, but the first clinical evidence for its efficacy was 2010. Liping Chen identified PD-1 as a target for immunotherapy in 2002. Those papers that published that it was true with 2012. And what we are in the midst of wondering whether or not we will have any clinical evidence for efficacy. But again, we started on this pathway in 2010. Okay, well, I think Bob will go next, and then we'll have questions after that.
Thank you, Doug, for a very exciting presentation, and we're all looking forward to hearing how these studies go, and hopefully in a very positive way in the next few years. Our next speaker is Professor Robert Maki. Bob received his MD and PhD degrees from Cornell University Medical College. He has been on the faculty of Memorial Sloan Kettering Cancer Center, Weill Cornell Medical College, and Mount Sinai Medical Center, where he was a professor and section chief of pediatric hematology oncology. Currently, he is a professor of medicine at Hofstra Northwell Medical School and director of experimental therapeutics at the Don Monte Division of Medical Oncology and Hematology at Northwell Health's Monter Cancer Center. He is also a professor at Cold Spring Harbor Laboratory. Bob's specialty is in the area of sarcoma, where he is a world-leading clinician, and he has been involved in clinical trials, including immunotherapies and new drug development. Bob has received numerous awards, including being a fellow of the American College of Physicians, and he's been named Clinician of the Year by the Life Raft Group. It's my pleasure to introduce Dr. Robert Maki.
Thanks, everybody. Thanks for the opportunity to speak tonight. Thanks for coming out to hear a little bit about cancer and immunology. I'm going to reiterate a lot of things Doug was saying as well, but coming from a different perspective. And what I'd like to do is get after some of the issues that I deal with as a clinician as well every day. I'm going to actually discuss that a little bit first in terms of immunity and what it means to have immunity against infection, against a cancer. I do have my disclosures. I do consult for a number of companies, and we do research with these people. And just so you have an idea of the different companies that I do work with. But the big question I'm always asked, I get this at least twice a clinic, a couple times today in fact, is how can I boost my immune system? And pretty clearly the answer is you just call a cardiac surgeon. You know, Dr. Oz will be happy to tell you all the good things that you can do to boost your immunity. And really it's a matter of definitions when it comes to all of these things. And just your ability to fight off illness, I usually refer to that as your constitution. You're somebody who just doesn't get ill very much, or you're always somebody who has a runny nose or something like that. That's these cellular barriers to infection, our skin and the mucus lining of our gut and up here in our sinuses, all of those things, stomach acid, all these things help prevent us from getting infections. And another group of cells that don't really have a specific way of attacking an infection or let's say a cancer, these are cells that are involved with what's called innate immunity. And these respond very quickly to infections that are trying to bother us. What Dr. Fearon was discussing though is the adaptive immune system, which involves both these T cells that you saw attacking a cancer cell, as well as other cells such as B cells, which make antibodies in your body against infections and allow for this memory response to reside in your body in case you recognize and get exposed to the same infection again later. And really to fight off infection well, and technically cancer as well, you need both of these things to work well. Otherwise, you're going to have a problem and get infections. That is a defective immune system. And we see this, people have these genetic syndromes where one thing is missing or another, and they are predisposed in particular to infections. What are the cells that are involved in this process? These are many of the white blood cells in our body. These are all made by the bone marrow, and they circulate around or reside in the different tissues of our bodies. And some of them have this non-specific function where they're trying to kill off things as they're trying to invade your skin, like a bacteria or a virus, or things that float around our bloodstream, the B cells and the T cells here on the right, which are responsible for infections and watching things that actually make it into the interior of the body. Once the infections have breached these initial forces that are trying to prevent us from getting infected. All right, and so the key cells that are involved here are the B cells, again, which make antibodies, and the T cells, like the cytotoxic T lymphocytes, which can kill cancer cells, as you just saw, and also kill virus-infected cells. All right, so how do you boost your immunity in terms of the innate immunity? How do you make those non-specific cells work better? How do you improve these barriers? These are things that we really don't understand very much. Certainly probiotics and yogurt. If you've gotten exposed to, taken a series of antibiotics, you completely change the bacteria that are in your gut. Perhaps that's the sort of thing that you need to help protect you against infection in the future, but we really don't understand the details of that. One thing I will recommend is that a lot of people do take supplements of some sort. Memorial Sloan Kettering have put together a very nice website called About Herbs, and this can tell you a lot about all these natural products that people are taking and what is purported to be useful about them, and what the actual data are regarding that process. So, for example, for ginkgo biloba, it says ginkgo does not improve memory or cognitive functioning in healthy people, and it goes into the details based on surveying the literature for each of these compounds. So if you have a question about CoQ10 or whatever the supplement may be, this is really a nice resource, and it's been curated over the last 10 or 15 years. They've done a really nice job with it. In terms of adaptive immunity, what can you do to boost your immune system? Well, is vaccination. And we talk about using hepatitis B vaccinations to prevent you from getting hepatitis B, which leads to cirrhosis, which leads to liver cancer. But the vaccine that we have access to now that we should be getting as young people, kind of between age 10 and 13, is the good old HPV vaccination, because human papillomavirus is responsible for not just cervical cancer of the uterus, but also for an increasing number of throat cancers as well as penile and anal cancers. Now, just about everybody who's sexually active has HPV, so if we're going to make a dent in some forms of preventable cancer, this is one way to do that. So this is one way to technically boost your immunity against cancer. So in terms of the approaches that are used today against cancer, these are some of them, and Dr. Fearon already discussed these to some degree. We've been trying to use cancer vaccines for a very long time. They really have not been very effective, and they're largely experimental. Perhaps the one exception for this is this Provenge vaccine, where you take some of the white blood cells out of your body, you train them to recognize proteins that are found in prostate cancer and give those cells as a vaccine back to people. It helps a little bit, but profoundly expensive, over $100,000 for 3 vaccinations, and the benefit that people get, it's relatively marginal. Antibodies, these are made by the B cells, the special white blood cells. This technology, after having recognized the structure of these proteins and what they can do in the body against infections, especially bacteria, we now use these very specific tools as cancer drugs. And it's probably the most commonly used approach in cancer now, beyond the regular toxic chemotherapy medications themselves. Immune checkpoint inhibitors, you just heard Dr. Fearon discuss these. I'll talk about these a little bit more, as I will discuss some of these CAR T cells, chimeric antigen receptor T cells, genetic therapy that has been discussed in the New York Times and on CBS Sunday Morning. There's this past weekend as well, some of the new approaches that are being taken to trigger an immune response against cancer. A lot of these do involve T cells, to be sure. Provenge eventually does stimulate T cells to attack prostate cancer. These immune checkpoint inhibitors stimulate T cells that were otherwise being too lazy and not activating themselves against the cancer cells if they were there already. And these CAR T cells are engineered versions of T cells that can recognize and kill cancer cells by themselves. I wanted to show you a little picture here. This is just a molecular structure of an antibody molecule. And this structure, which has places for proteins to bind on in 2 places in this particular version of an immunoglobulin, this is the sort of structure that's been used to raise basically vaccines, antibodies against a variety of proteins. And these proteins are found on tumor cells, and then we give these antibodies, which are schematized here. They recognize the protein of interest. So for bevacizumab, this is something called VEGF, vascular endothelial growth factor, something that prevents, this antibody then would prevent the growth of blood vessels into tumors. Then you also can starve tumor cells in different ways by blocking special signals that keep the cells alive, whether they are solid tumors such as breast cancer or things like lymphoma, in which rituximab is an active compound. So these are exquisitely specific molecules. They don't bind to other molecules, even if they have a very small genetic difference. And as a result, they become really useful tools for cancer, because the side effects are something that we can control much better than we can with the regular chemotherapy drugs. So very, very nice approach, and this is commonly used now for cancer therapy. As Doug was getting at before, what do these immune checkpoint inhibitors do? They basically take the brakes off the immune system. And they're relatively nonspecific because these same molecules which turn off immune responses are found on both the cells that might fight a cancer, as well as those that fight infection, as well as those that might, just by happenstance, may attack a normal organ of your body. So good immune responses, if you get an infection with flu, let's say, you have this upregulation, a larger number of T cells that recognize the flu virus and its proteins and kills off the infected cells, but then you want those things to calm down and go away, and that's where these molecules exist in T cells to turn off an immune response. If they stay too active, well then you end up with autoimmunity, where the body's being attacked by these T cells that are chronically turned on. So it's important for T cells to be both turned on and turned off. So in either one of these, if you have too much of one or the other, it's not good. Anergy, where you don't get their immune response at all. This is what viruses have been trying to do throughout evolution. So EBV, Epstein-Barr virus, which causes mononucleosis, it's engineered itself. It's been selected against being seen by the immune system. It's really very much a stealth virus that has many ways of preventing the immune system from seeing it, for example. So these are, either of these are counterproductive. What we want are these responses that you can control, and the immune checkpoint inhibitors do give us some degree of control that we've never had before for cancer therapy. So if you've been unfortunate to watch commercial TV news, you've been assaulted by commercials for, this is a bizarre one, irritable bowel syndrome and diabetes and arthritis and, well, you know what. But now, and I'm sure you've seen the advertisements now for these direct-to-patient ads for treatments for lung cancer, nivolumab or Opdivo, which is approved for, I'll show you some of the diagnoses for which it's now approved, or to give it equal time, the other big drug that's used most frequently these days, pembrolizumab or Keytruda. And as Doug was referring to already and has discussed, how do these drugs work? Again, you're taking the brakes off the immune system. Normally, the immune system is turned on and then turned off. Again, these T cells are turned on and then turned off. And in fact, the tumor cells have the means of turning off that immune response by the presence of these special molecules. They figured it out that you want to negate or neutralize that immune response. So by taking away that negative signal, it's sort of a double negative. By taking away the blocking response, taking your foot off the brake, you allow the immune response to go forward. But again, it's a nonspecific event. It turns on every T cell in the body, which is great if you're trying to fight cancer, but bad if you have Crohn's disease, let's say. All right. Very, very nice graphic. Anyway, so I'm going to show you another thing that make you a bit of a clinical statistician as well, and we're going to review a few of the clinical trials data. This requires an understanding of these so-called survival curves. These are morbid actuarial devices that allow us to discuss who's doing well and who's not, basically who's alive and who's dead. And as you see time pass, well, things happen. If everybody survives just as may happen in a normal population over time, then you'll see no change in this fact that 100% of people are still alive. If everybody dies from something really bad, like an advanced cancer, you'll see this curve crash down to the 0 point. And if people have metastatic cancer, people die at some rate, and we'll see that over time, some number of people are still alive. And we compare these curves to determine how good one treatment is versus the other. And this is the bread and butter of randomized phase 2 and phase 3 clinical trials, which have led to all the advances, all the approvals of the drugs we use. So for some examples of that, one of the first ones, it doesn't look that impressive because there isn't that much space between the curves. This was as Dr. Fearon was mentioning a little while ago, melanoma. These were the first people to show signs of benefit from one of these immune checkpoint inhibitors. Jim Allison and the discovery of the CTLA-4 molecule led to the development of ipilimumab, also called Yervoy. And this was the first study that showed that immunotherapy with chemotherapy was better than just giving the chemotherapy alone. Not a huge difference, but you can say that it is statistically different from the two, and this led to the approval of ipilimumab based on work that was done in the late 2000s and early 2010s. There was more impressive data as we went along. So here's, instead of a CTLA-4 inhibitor, here's a PD-1 inhibitor. This is nivolumab, and this clearly in melanoma patients who had recurrences was better than dacarbazine, the plain old chemotherapy. And in fact, if you look out here, you see that this curve isn't going down to zero. That means people are staying alive longer and without disease. This, as people stay on this and this curve stays flat, that's what we start calling remissions or cures. So this is exactly what we're looking for, and this was really quite striking to see this happen for the first time. Melanoma is sort of the poster child for these diagnoses in terms of the benefit, but it still isn't everybody, and Dr. Fearon gave some really good reasons why these drugs may not work, from the lack of penetration of the T cells into the tumor or other reasons to reject the T cells from being active within the tumor. Now, again, melanoma is the poster child. Here's data from lung cancer, and this is one of the most common cancers. It's the most common fatal cancer in the United States. About 175, 180,000 people get this a year, and it's the #1 killer in terms of cancer in the U.S. And the data are not as impressive. But this was still a relatively revolutionary study in that for this very common cancer, the community showed for the first time that immunotherapy with this PD-1 inhibitor was better than giving chemotherapy. So it immediately moved immunotherapy into first place, and it's used now more and more earlier in the treatment of people with cancer so that you can avoid some of these poisons which cause all kind of side effects. Yes, there are side effects with these as well, and they can be difficult to manage, and perhaps something we can discuss during the discussion. I'd like to just outline now, just in the past few years now, all the diagnoses that have approvals for one or another of these so-called immune checkpoint inhibitors. And so a number of diagnoses, we're happy to have these drugs available, and we use these routinely now in the clinic. We're trying to build on this now. Can you combine these drugs? Do you combine them with chemotherapy? What new things can you add to them, such as Dr. Fearon's study with the cytokine inhibitor or chemokine inhibitor. But there are a number of diagnoses also, as he pointed out, that are not responding. And in fact, most people with a number of these diagnoses are not responding for a long time. So we definitely need some improvements, and we're hopeful that the work with Dr. Fearon will lead to things that we can do here within our own healthcare system as well, right here on Long Island. We've got some of the brightest lights here in cancer research, and hopefully be able to apply those to trials. Now, the next thing I'd like to mention here in terms of these immunotherapeutics, if immune checkpoint inhibitors, which you've seen now approved for these different diagnoses, are the shotgun, well, the more specific tool that we have are these so-called chimeric antigen receptor T cells. And why is it more specific? Well, with the immune checkpoint inhibitors, you don't know the target, whereas with the chimeric antigen receptor T cells, you actually engineer these things, and you know the target and the context in which the molecule is found very, very well. So how do you cook up one of these things? Well, it actually has been trial and error, and Carl June and others down at Penn and groups at the NCI have really led the way in terms of figuring out how to do this. It's engineering a tour de force, and it involves taking out blood cells from your body. You can take out the whole blood volume and spin it down, collect the blood cells, and give the red blood cells back to a person, then just collect the white blood cells. You take those and you select out the key cells that are the T cells, and you now infect them. It sounds anathema that you'd actually use a virus to treat these cells, but this is how you introduce a special virus which now has basically the trigger to be pulled when the T cell encounters a cancer cell. And you then give a patient some chemotherapy to clear out their own immune system, and now introduce these special engineered T cells, and then you can watch what happens both in terms of blood tests that you do for the patient, looking for the presence, how long do the cells last in the body, and you can see if they actually do something in terms of the number of cancer cells that remain in the body. Some details in terms of what this virus entails. There are basically ways of activating the T cell in a very methodical way against a very specific molecule, the CD19 molecule. This is the CAR T cell that's just been recommended for approval by the FDA. And this recognizes the CD19 molecule. CD19 is a molecule only found on the B cells, and it turns out that acute lymphoblastic leukemia, ALL, myeloma, lymphomas, those sorts of different versions of chronic leukemias, all have this molecule on there, and therefore this kind of an approach could theoretically be used for any tumor that has a CD19 molecule on it. It's been tested, and this approval will revolve around acute lymphoblastic leukemia, where basically kids and young adults who most commonly get this, who had exhausted double bone marrow transplants and multiple rounds of chemotherapy, were treated with these drugs, actually cells, and had remissions once again, where they had, yeah, it does sound complicated, right? And it is, and it really is. This could be a limited number of centers that are able to use these sorts of therapies, but when they're effectively used, there is a very significant survival rate. This is the initial publication of the CAR T-cell data back from 2014. Very small number of patients being treated because of all the engineering that's required. It takes a small army of people to grow these cells, infect them, get them in enough numbers where you can give them back to a patient to be an effective, essentially, drug against that cancer. But nonetheless, it's kind of riding a bucking bronco because of all the side effects that happen with these things as well. So fine, it's an effective technology. Why don't we use it for everybody? Well, number one, you have to know the target. We know the target is CD19 on ALL. We don't know what the target is necessarily on, let's say, a pancreas cancer cell. Or we may have some idea, but that molecule might be found both on the pancreas cancer cell and also in the normal pancreas. Maybe it's also in the normal colon or in the normal stomach. Well, you don't want to start destroying the normal colon and the normal stomach along with the tumor, so you have to be very careful about the molecule that you choose to target. So for example, that speaks to one of the side effects of CAR T-cell therapy. If you wipe out every CD19 positive cell with this therapy, you have no more B cells, and you no longer make antibodies, and you need to get lifelong intravenous immunoglobulin to help fight off infections. Otherwise, you get bad bacterial infections. But it's a price to pay for being cured, hopefully, of your illness. And you also have to know the context in which these different molecules are bind. If you're looking at a bit of a protein, it has to bind to one of 6 adapter molecules that are found on every cell of the body. And if you don't have that adapter molecule in combination with the right protein, well, the CAR T-cell is never going to fit. It's never going to be able to do its thing. It doesn't recognize the target. And that does require a lot of work from our basic and translational immunologists to find out what these adapter molecules are sticking to and how we can find bits and pieces of different molecules that would be effective targets. And also, the treatment is very toxic. You do need ICU, intensive care unit level care for the shock sort of syndrome, very low blood pressure that people get for a prolonged period of time, for at least several days after exposure to these CAR T cells from this massive release of hormones, cytokines as they're called, that are released into the bloodstream. They also see problems with delirium, confusion, encephalopathy. Some people can lapse into a coma. Thank goodness it's just a temporary thing. They do come back, but it's certainly a scary ride for people to have to go through. It doesn't happen to everybody, but as many as about a quarter to a third of people are getting very severe side effects with the application of these cells. So the idea is to try and treat people when they don't have such large number of cancer cells in their body, try and get them down in terms of the numbers of the cells, and then apply this very aggressive approach to the patient. And it's also laborious. You have to find these new targets. It does take a lot of work, and to prove that there's specificity on one cell, that the molecule is not found on another important cell of the body, brain, heart, whatever it may be, that's critical before you move forward with this kind of an approach. And there's some other ways of trying to get around that problem as well, about the specificity issue of using less specific but still active killing cells of the body, such as the NK cells, natural killer cells. These also have the potential for killing cancer cells in a non-specific way, and maybe another approach that's taken in the near future. In fact, they're already doing studies of this at several centers in the US and worldwide. The last and most another important point is that it's not going to be cheap. So basically, these antibodies run about $10,000 every 3 weeks. So $170,000 per year per patient. So that benefit better be pretty big for you to justify using these drugs. Well, I mean, it's a free country. Everybody gets access, at least on Medicare, let's say, to these compounds if they are approved. But other places, like in England, they say, okay, well, if treatment's going to be, several $1 million and it only prolongs life by a few weeks, it's certainly not going to be valuable. Well, we have to make this decision as a society as to what's going to be worth it and what's not. So yeah, who knows what the going price is going to be for this very newfangled and technically complicated approach will be. So something to keep in mind, we're going to have a limited amount of these things we can actually use. At some point, we have to choose which ones we move forward with carefully and hopefully find the ones that work the best and give us the best bang for our buck, literally and figuratively. So immunotherapy now is a new way to treat cancer effectively and alongside of the other treatments we use now, which are surgery and radiation therapy and chemotherapy. Not all types of cancer respond well. Sometimes it's a matter of not getting the T cells over there to the tumor, and we'll hopefully see that some of these approaches do bear fruit, both in terms of what he's studying this microenvironment of the tumor, in other words, better, looking at different combinations of immunotherapies and new agents, and then also trying to find new targets and new diagnoses for these technologies as well. And with that, I'm going to stop with a picture of this very nice plant which gave rise to something we call acetylsalicylic acid, or aspirin. Natural products still have a place in our armamentarium against disease as well. So with that, I'll stop, and I think we'll move forward with questions.